Let's discuss on hemoglobinopathies. Hemoglobinopathies are a group of disorders due to the alteration in hemoglobin structure. That is the qualitative changes. The quality of work or functions of hemoglobins are altered. That is the qualitative changes or impaired synthesis of polypeptide chains. So polypeptide chains in the form of alpha or beta chains. So due to some mutations, the impaired synthesis of these chain happens, leading to the structural as well as functional alterations in the hemoglobin structure. So all these come under the category of hemoglobinopathies. So abnormalities in the primary sequence of globin chain leads to the hemoglobinopathies. For example, sickle cell anemia or hemoglobin S. So abnormalities in the rate of synthesis would result in thalassemia. In other words, the normal globin chain in abnormal concentration results in thalassemia, like beta thalassemia or alpha thalassemia. So in these beta thalassemia, the proper synthesis or insufficient or deficient synthesis of beta chain happens. So hemoglobinopathies may be congenital or acquired. So let us see how it is being the various type of pathies have been divided on the congenital and acquired categories. So congenital, in the congenital category, there are two types of disorders. One is the structural hemoglobinopathies and second is the thalassemias. So in the structural hemoglobinopathies, they basically happens due to some point mutations, alteration in the amino acid sequence. So altered amino acid sequence of globin chain happens due to mutations. Most commonly, these are the point mutations. So in this category, hemoglobin S, hemoglobin C, and hemoglobin E are most common. Then another type is thalassemia. That is a type of quantitative disorder, quantitative changes in the polypeptide chains. So this is due to the defective biosynthesis of globin chain. They can be alpha thalassemia or beta thalassemia. In alpha thalassemia, there is inefficient or deficient synthesis of alpha or beta chain. Then acquired, in the acquired category, the reason can be the toxic exposure, maybe due to the exposure to carbon monoxide or sulfated atmosphere or due to the overexposure to carbon dioxide. So these lead to the derivative in the form of methemoglobin, sulfhemoglobin or carboxyhemoglobin, etc., which have lost the capability to carry the oxygen. So here we will see in this table the altered type of hemoglobins, like hemoglobin S, hemoglobin C, hemoglobin E, and hemoglobin D, and all these are altered kind of hemoglobins, and these generally happens due to the point mutations in the genes. So we will see the HBS. So it happens in the beta chain, and at the sixth position in the beta chain contains around 146 amino acid, and the sixth position amino acid, that is the glutamic acid, is being replaced by valine. And it actually happened at the level of gene, because and due to that the codon have been changed from GAG to GUG. So single base pair alterations from A to U led to the change from glutamic acid to valine. And similarly, in HBC, the glutamic acid has been replaced by the lysine, and the codon has been changed from, this is due to the codon change, that is from GAG to AAG. Similarly, in case of HBE, the beta 26 position amino acid in the beta chain, so that is the glutamic acid, is replaced by lysine. In HBD, the beta 121 amino acid, that is glutamic acid, is changed by glutamine. So then HBM, the proximal or distal histidine in alpha or beta chains, they have been replaced by, the histidine has been replaced by tyrosine. And these proximal amino acid directly, coordinate with, they participate in coordination bonding with the iron. So replacement with the tyrosine will mostly affect the coordination attitude of the hemoglobin, and that will alter its no oxygen carrying capacity also. So these are the molecular differences in HbA, HbS, and HbC. So in HbA, this is the adult human uh, hemoglobin. So there is a glutamic acid at sixth position, which is being replaced by valine in sickle cell anemia or HbS trait. So this is being replaced by valine. Similarly, in the HbC, that is the Cooley's hemoglobin. So replacement has been happened by lysine in place of glutamic acid. Now let's talk on thalassemia. Thalassemia is a group of genetically transmitted disorder of hemoglobin synthesis, and due to lack or decreased synthesis of alpha or beta globin chain. So because the synthesis of one of the globin chain is reduced, there is relatively excess synthesis of other global chain. So these globin chains may precipitate in the cell, causing the hemolysis, resulting in the hypochromic microcytic anemia. So all cases of thalassemia are characterized by deficit of hemoglobin A synthesis. So there will be nucleated RBCs in the peripheral circulation, and there are two major type of thalassemia. One is the alpha thalassemia and second is the beta. Alpha means the defect lies in the alpha chain. For beta means the effect lies in the beta chain. So alpha thalassemia, in this class, the synthesis of alpha globin chain is defective. And alpha globin chains are coded by 4 copies of alpha globin genes. So alpha thalassemia results from genetic defect in one or more copies of alpha globin chain, and is characterized by either decreased or total absence of synthesis of alpha globin chain. So the alpha thalassemia is again of 4 subtypes. One is the silent carrier type of alpha thalassemia. So only one of the 4 copies of alpha globin gene is mutated. Rest are normal. So such patients do not show any clinical symptoms, and they spend a normal span of life. Then alpha thalassemia trait. What is a trait? The 2 of the 4 copies of alpha globulin gene are mutated. They usually have only mild anemia and it's not fatal. So it is a heterozygous condition, and in which there is no expression of symptoms. Patient can survive with mild anemia. And the hemoglobin H disease, 3 in this category, the 3 of the 4 copies of alpha globin genes are mutated, and they have moderately severe hemolytic anemia. Out of all these, the fourth one is the worst condition, and its fetal state. And in this, all 4 copies of alpha globin genes are mutated. All mutant genes are there, and the resultant this is a fatal condition, lethal condition. Most of the affected are stillborn, that is the death before the birth, or die soon after the birth. So beta thalassemia, in beta thalassemia, the beta globin chain is impaired due to the genetic defect in the beta globin chain. So beta globin chains are coded by 2 copies of beta globin genes, and is characterized by decreased or total absence of synthesis of beta globin chain. It is again of 2 subtypes. One is the beta thalassemia minor, that is actually a trait, And then second is the beta thalassemia major. So in beta thalassemia minor, one of the two copies of beta globin genes are mutated. It is again an heterozygous condition. So the presence of one of the normal gene in the heterozygous allow enough normal globin chain synthesis so that affected individuals are usually asymptomatic. So they spend normal lifespan. Then individual may be completely normal or have mild anemia. Then beta thalassemia major. In this category, this is a homozygous condition, means both carrying the two mutated beta globulin genes. Means from the offspring has derived the two genes, both are affected genes from one from each parent. So this is a homozygous state. And in this this thalassemia major leads to severe anemia, that is a hypochromic microcytic anemia. And there is a deficit of hemoglobin A synthesis, and RBCs are generally nucleated. You can see the picture of nucleated hemoglobin. And in the peripheral circulation, homozygous beta thalassemia is characterized by severe anemia, hypersplenism, and hepatosplenomegaly, because there is a mass destruction of RBCs. They are functionally impaired. They lost the capability to carry the oxygen. And X-ray shows one more important finding associated with beta thalassemia major is that there is an expanded X-ray shows the expanded marrow in the skull bones, producing hair on end appearance. So these spicules like appearance perpendicular to the surface of this bone, skull bone. So that kind of images, abnormal images can be shown in the X-ray um, pictures. Then treatment, the repeated blood transfusion is the treatment, and this may lead to the iron overload. Bone marrow transplant is another way to treat the patient. Then in sickle cell anemia, let's discuss in detail about the sickle cell anemia. So what happened in sickle cell anemia? I've already told you that at the sixth position there is a glutamic acid which is being replaced by valine. There is a mild shifting of these number. This is actually the sixth position, sixth number amino acid, that is glutamic acid, and which is being replaced by valine. So as polar glutamic acid residue is replaced by the non-polar valine, it generates hydrophobic contact point called the sticky patches. So because of these, the primary, you can here better understand the importance of primary structure of amino acids of the polypeptide chain. So just one polar amino acid has been replaced by valine, which is totally non-polar. So that will accordingly alter the secondary and tertiary and all these higher level of organization of protein structure. And the resultant polypeptide chain is having the sticky patch onto the outer surface of the beta globin chain. So beta 1 and beta 2 chain both have hydrophobic patch of deoxyhemoglobin S. So they lost, again they are not able to carry the oxygen because of these alterations. And this alteration markedly reduce the solubility also of the deoxygenated hemoglobin S. So when hemoglobin X is deoxygenated, the sticky patch can bind to one another. They are sticking to each other, and this binding causes the polymerization of deoxy HBS, forming the insoluble long tubular fiber. So tubular fibers formation happens in the HBS, and these insoluble fibers of deoxygenated HBS are responsible for deforming the red blood cells, which look like a blade of sickle. And this sickling is because of this sticking and polymerization of these hemoglobin S, leading to these long fiber formation, insoluble fibers of deoxygenated hemoglobin S. And resultant alteration in the structure of RBCs will lead to water losing, lead to the loosening of water. Like water retention capacity of RBC is lost, and the RBCs become more fragile and have a much shorter span of time than the normal cell. Normally it is 120 days, but it is now reduced to 70 days only, and leading to the lysis of the red blood cells, and this results in hemolytic anemia called the sickle cell anemia. So frequent no breakdown of RBCs leading to, and there is scavenging all being, there is an important role of spleen. That's how there is an there is a splenomegaly, very marked splenomegaly is there. Then the more serious consequences is that the small blood capillaries in different organs become blocked by the long abnormally shaped RBCs, because you can see these are the normal RBCs, but due to the sickling, due to the shape changes in shape, they do occlude the passage of the small capillaries, leading to the blockage, and this interrupts the supply of oxygen and leads to anoxia to the site, and there is an oxygen deprivation which causes pain and eventually death to that those tissues or cells where this supply was there. So this is the major manifestation of this sickle cell anemia. Okay, and it is characterized by chronic hemolytic anemia, tissue damage and pain, and increased susceptibility to infection. And such patients usually die in the adult age. So this sickle cell anemia, there is one advantage also there with the patient suffering from this sickle cell anemia, like person with sickle cell trait or disease are resistant to malaria caused by plasmodium falciparum, because half of the these uh, this sickle cell in the sickle shape, these RBCs, their water retention capacity is lost, and they become more fragile and have much shorter span of time than the normal cell. Normal cells have 120 days lifespan, while the sickled cells have the time reduced to 70 days only, leading to severe lysis of these RBCs and result in hemolytic anemia called the sickle cell anemia. And that's how for scavenging of these RBCs, no shape of spleen is raised, and there is a severe, splenomegaly is there, because it is one of the important organ of reticuloendothelial system. It is meant to scavenge the dead and decayed RBCs. So that's how there is a marked splenomegaly is there. And moreover, what is happening here, the more serious consequences is that the small blood capillaries in different organs become blocked because of very peculiar and altered shape of RBCs. So they are not easily passed through the passage, and this interrupts the supply of oxygen and leads to anoxia to the tissues or the cells where the supply was going on. So which causes severe pain and eventually death of the cell. And this leads to the patient is suffering from chronic hemolytic anemia, tissue damage and pain, with increased susceptibility to infection as well, because all humoral component of immune system, they are in the blood, so blood supply is not proper. This lead to the you know uh, impaired defense response, because the cells are not able to reach to the defense cells are not able to reach to the site of infection. Then the sickle cell anemia and the sickle cell trait, what is the basic difference in between the two? So sickle cell anemia is a homozygous disorder in which the individual has inherited the two mutant globin genes, one each from each parent. and it is characterized by hemolytic anemia, tissue damage, and pain. So there is a total expression of symptoms of the disease, and such patient they do not survive long, and they die in maximum in the adult age. And secondarily, the sickle cell trait is an heterozygous condition in which one of the gene is only affected and another gene is normal. So two parents, one is suffering from the disease and another is free. So the offspring has has the gene, one he is derived from the mother and one from father. So one is affected, one is normal. So this condition is heterozygous state, and the patient is living a normal life with no expression of symptoms. So this is, though patient is straight, further progenies can be affected if they are come in the homozygous condition. Otherwise, the patient is living a normal span of life except for mild anemia. One major advantage of the patient suffering from sickle cell anemia is that person with sickle cell trait are resistant to malaria, which is being caused by Plasmodium falciparum. And high incidences of sickle cell gene in population coincides with the area endemic with the malaria. It has been found. Then HBS affords protection against Plasmodium falciparum infection, and hence the abnormal gene was found to offer a biologic advantage. This is called an what a term we can use for it is balanced polymorphism. So that's all about the various types of hemoglobinopathy.